B cells, as any other cells in our body, are produced from stem cells. This process occurs in the bone marrow, where from stem cell we make lymphoblast. Once lymphoblast is produced, then lymphoblast mature into naive B cell, and naive B cell is able to leak from the bone marrow into the blood. From the blood, naive B cell goes into the lymphoid organs. In adult person, the major lymphoid organs are lymph nodes and spleen. So let's take spleen as a lymphoid organ. When naive B cells enter into the spleen, basically they begin to wait for the activation, and activation of naive B cells is triggered by antigen. Let's take for example Staphylococcus aureus. When this bacteria leaks into our organism, in response to antigen, our immune system activates naive B cells, and there are two possibilities. Some naive B cells differentiate into short-living plasma cells, which are the plasma cells that produce immunoglobulin M antibodies straight away. The positive aspect of short-living plasma cells is the speed. These cells react very quickly, and because of this, they provide initial immune response. But the drawback is that they produce immunoglobulin M antibodies which are non-specific, and this makes them not very effective. So, to make a more effective antibodies, some amount of naive B cells differentiate into centroblast. The unique feature of centroblast is their ability to undergo somatic hypermutation and class switching. Now we have to understand the purpose of this process. To explain this, here we have bacteria. Bacteria consist of some bacterial antigens. Let's mark bacteria antigens with green and yellow colors. In response to bacteria, centroblast in lymphoid organs trying to find the best immunoglobulin to kill this bacteria. B cells can produce a great variety of immunoglobulins, but the problem is that they do not know yet which immunoglobulins they need to kill the bacteria. Recall that immunoglobulins consist of immunoglobulins heavy chains and immunoglobulins light chains. The genetic information how to make immunoglobulin heavy chains is located on chromosome 14. Let's mark the genes that participate in heavy chains formation as V, D, G, and C genes. The genetic information how to make light chains of immunoglobulins is located on chromosome 22. Let's also mark these genes as V, D, G, and C genes. So, how will be looking the DNA molecule of a perfect B cell to kill this bacteria? B cells have to find the sequence of genes that will ideally suit this bacteria. In this case, for immunoglobulin heavy chain, it's V1, D1, G2, and CD. From this gene sequence, B cells will make a heavy chains. The same process occurs in a light chain gene, where a cell step by step selects the most suitable genes for this bacteria, and this results in formation of a gene sequence from which B-lymphocytes will form a light chains of immunoglobulins. So this entire process in the end causes formation of a B cell that makes immunoglobulins with very high affinity to this bacteria. And exactly these highly selective immunoglobulins can very efficiently kill this bacteria. But because B cells cannot select the specific gene sequence straight away, this process results in formation of a multiple broken B cells with low affinity to this bacteria. And in fact, before B cell will find the right gene sequence, during this process we produce a huge amount of broken B cells. The major problem with broken B cells with low affinity to bacteria is that they cannot kill the bacteria. They are totally useless, and nature is absolutely merciless to useless stuff. Broken B cells with low affinity to antigens undergo apoptosis, or we call this negative selection, that results in their death. In contrast to this, B cell with high affinity to bacteria undergo proliferation that results in formation of a multiple identical B cells with high affinity to bacteria. Basically, they are clones. High affinity B cells produce high affinity antibodies, and such antibodies will very quickly kill the bacteria. So, somatic hypermutation and class switching cause production of a massive amount of broken centroblast with low affinity to antigen. Such cells undergo negative selection that results in their death. But also, somatic hypermutation and class switching produce centroblast with high affinity to antigen. These high affinity centroblasts differentiate into centrocytes, and from centrocytes we can form memory B cells that remain in the spleen, and also we can form long-living plasma cells that will enter into the bloodstream. As we can see, cells are located in different areas of the lymph nodes or spleen, and these areas in lymphoid organs we call zones. We have marginal zone where memory B cells live. We have mantle zone where naive B cells live. We have germinal center that has two zones. In the dark zone live centroblast, it's zone where somatic hypermutations and class switching occur. And also we have the light zone, where centrocytes live. Why it's important? Because we have various malignancies where a particular subtype of lymphoid cells cannot die or hyperproliferate. As a result, the amount of cells increase. If cells accumulate in the blood, we call this leukemia. If cells accumulate in the tissues as lymphoid nodes or spleen, we call this lymphoma. And with increasing amount of cells, the size of the zone where they live increase. And lymphomas are named according to hyperextension of a particular zone. If mutation inhibits apoptosis of naive B cells, the amount of B cells in the blood increase, and this condition we call chronic lymphocytic leukemia. Some mutations cause immortality of centrocytes. The amount of centrocytes in the germinal center increase, and this causes hyperextension of the germinal center light zone, and exactly this happens in follicular lymphoma. Some mutations cause pathological increase in amount of centroblast in the germinal center dark zone. This happens in case of Burkitt lymphoma and diffuse large B-cell lymphoma. In both these conditions, we can detect hyperextension of the dark zone due to the increasing amount of pathological centroblast. Some mutations cause increasing amount of naive B cells, and because naive B cells live in the mantle zone, this causes hyperextension of the mantle zone, and this condition we call mantle zone lymphoma. Some mutations cause increasing amount of memory B cells, and because memory B cells live in the marginal zone, this causes hyperextension of the marginal zone, and this condition we call marginal zone lymphoma.